Hey guys, so I'm Pratham Bawali and I decode how the world really works. So, in this session, we will be knowing how a drug is born from bench to animal, then clinical trial, then the FDA approval. So, basically, the journey of science, failure, hope, and breakthrough. So, in this video, we will be learning the complete overview, the foundation from where the drug is born to where how the patients take it. So in this session, the first thing we will learn is the beginning of the discovery. The discovery from there is a starting point from where the drug is actually take place. So here we can see every drug start with a question, not from a formula, but from a question. It begins with identifying a disease mechanism and finding a target to attack. And where the scientists search for biological agents that can interfere, block, kill, or correct the malfunction which is taking place in the cell, in the organ, in the tissue, in the blood cells, and anywhere in the. So basically, we can say a drug is born, not in factories, but in imagination. Then the very second thing what we are doing is identifying the biological targets. What are the biological targets? Here, the criteria we will see. The criteria for identifying the biological target is scientists must choose a specific disease pathway or receptors which are already built in a patient, and these targets must be druggable. Like any molecule, any new chemical entity or any chemical entity should cure it, should block that receptor, should block that pathway, should change the interaction between anything. So it is like that. This targets must be druggable. That's the main point. And meaning it can be chemically modified and clinically relevant to the disease progression. So clinically relevant to disease progression means if you took a drug and it is not working, then there is no use of that drug. So it basically means that the clinically it should be treating your particular disease. In Now we will see a real world example, which is HER2 receptor in breast cancers, which the discovery of protein overexpansion led to the development of trastuzumab herceptin resolution treatment for HER2 patients, which is basically it stopped the development. The drug, the molecule completely stopped the development of herceptin, which is which is a pathway for developing of that cancer. Now, um, if you will know, the very beginning, it start with the molecule hunt. The molecule hunt, where it begins, if you, now it is a very crucial part because as you can see here, 1 out of 10,000 molecules survive. There will be more than 10,000 molecules, even there will be on lakhs of numbers. Here it is because minimum is 10,000 compounds. So searching libraries are like researchers screen over 1 lakh plus compound using high throughput automation. In today's generation, everything is focused on AI and machine learning. That's the reason modern AI and machine learning accelerate this by predicting binding affinity and potential toxicity. From thousands of hits, only a few become leads for the optimization, which means if you want to discover one particular drug molecule, one particular molecule, then you have to go through lakhs of compound which is suitable, and from that only one get the approval. That is where it takes place. Then lead optimization, which is basically what we are doing in this lead optimization. What we are doing, uh, medicinal chemists modify molecule to find the hero compound, which means the compound which is having the only potency, and it should be like maximizing the biological effect of the drug on its target. Toxicity is the one of the main reason, minimizing harmful side effects and off target interaction should be there, no toxicity should be there. And then solubility, ensuring the drug can dissolve and absorb by the body. That is very These three parameters are very important in lead optimization. If these three parameters will be not there, then your drug is not successful. Then preclinical or in vitro studies. What is preclinical and in vitro studies? It is basically, you will know the test tube worlds. It is a test tube world. Before touching a living organism, drugs are tested on cultured cells, enzymes, and bacteria. Here we can see IC50 value, half maximal inhibitory concentration. Then cell deaths percentage, we are which is effectiveness against the cancer lines. Then receptor affinities, how tightly it binds. Only safe and effective candidates move forward. Without in vitro studies, we can't even imagine how will the drug will survive in more. So these are the basic terms that you should know. And then the preclinical in vivo studies where the animal testing models are presented. In this, common models include mice, rats, zebrafish. These studies are critical for understanding how a complex living system processes the drugs. The 3R principles are replace, reduce, refine, guides ethical conduct. Key outputs parameters are like pharmacokinetics and absorption, distribution, metabolism, excretion. Then toxicity, LD50, the which is determining lethal dose limits. Organ impacts, monitor liver, kidney, and brain health. Effective dose like ED50, which is estimating the therapeutic dose. Then why animals are still used? This is a very major questions which now take place. If you will know now in US FDA has done a major uh, that no monkeys will be tested. They stop for testing on monkeys any particular drugs or any particular NC. But testing in monkeys, because for Homo sapiens, the very legitimate match is a monkey only. Then we will understand why animals are still used, because the basic thing is biological complexity. Human biology is too complex to simulate fully in a dish. An organism circulatory and immune system interact in way computer model cannot yet perfectly predict. Ethical ethical imperative, which is we must prove safety biological effects before risking a human life, which is more important, because at the end of the day we are making drugs for human products. The regulatory pathway mandate in vivo data to prevent catastrophic failure in human trials. Then preclinical approval packages, which is like to process the human trials, a company must file an investigational new drug, which is IND applications, which consist of comprehensive toxicity reports. Then pharmacodynamics model data. Then safety margin and risk analysis, and manufacturing and also the formulation reports. Then only the FDA will approve your the for preclinical studies. Then here we go for the phase one trial safety first, which is safety first. If you will know about phase one is basically safety first, in where healthy volunteers are taken, like 20 to 100 healthy volunteers are taken. Objective for this trial is it safe for humans or not? To identify that, we are doing this test. This phase is not about curing the disease yet. It focuses on finding the maximum tolerated dose (MTD), which is called, and observing side effects. The duration for this clinical phase is like 1 to 2 years, and the survival rate of the drug. Survive here we are talking about the drug, not about any patients, not about any disease or anything. We are talking only about the drug, and the survival rate for this stage is 70%. So which take the drug to move forward. Then phase 2. Phase 2 is like it take place in like 100 to 300 of the patients with the disease are treated. The goal is to balance efficacy, like normally you can know, like tumor shrinkage, symptoms relief with safety. Everything take place in safety. Here the percentage of chances of survival of a drug is 33%. Just 33% move to phase 3, and the rest of it is the failure rate, which is ineffective, unsafe. Here where the patients will get the symptoms or any safety if it is not matching accordingly. Then here we have phase 3. We are large scale proof we are taking. Participants will be around 1,000 to 3,000 diverse patients across multiple countries, and this is the final exam. Also, you can say the drug is compared against standard treatment or placebo to statistically prove clinical benefits. Comparison versus the existing drugs, solid risk, or like a which is this benefit ratio. Huh. Here the main thing, in large scale proofing, it costs around $500 million to $1 billion. This is just number, but in each and every cases has took place like more than 1 billion to 2 billion, 2 billion plus dollars. It is taking place. Now, if your drug survives in that, then NDA submission to FDA, which is a new drug application. It maybe consist of lakhs of pages of data. After a successful phase 3, the company files an NDA. This massive dossier includes everything from manufacturing SOPs to risk management plans and proposed labeling also. Then FDA review board evaluation, where the FDA assembles a specialized team to review every aspects of the application. The standard review time is 6 to 10 months. The verdict approval requires strong efficacy and acceptable safety margin, validation, manufacturing qualities. The review team will do the maybe if we are doing on oncology, then the clinical team must have a oncologist and medical doctors, pharmacologists, toxicologists, biostatisticians, and then manufacturing auditors. They all are having this. So in phase 4 is nothing but it's a post-marketing. Post-marketing, the journey does not end and approval. Companies must continue to monitor the drug in the real world. We're monitoring rare side effects, long-term safety effects, real-world effectiveness, expansions to new indicators, where the drug basically evolves. It is having its own product life cycle, where if you will initiate a product, then it is having a and you will initiate the product will be at medium range, it will be independent, and then it will be the decay phase where the product will no more use maybe the product will be in the market like for 20, 30 years, where it takes the, like, basically monitors everything. Then the final thing is, a drug is not a product. It is a scientific war won for humanity, born in curiosity, tested in sacrifices, approval in evidence.